This video is on Chapter 19, Part B, The Power of Genetics. In the first half, we give credit to Gregor Mendel and explain his findings and put them in perspective with current opinion in genetics. And in the second half of the video, we talk about some genetic techniques which are important. Genetics has evolved from a very primitive science to now one that demands respect. Genetics as a field of science has been around for about 150 years, but over the last few decades, it's really taken off in its own right. In the early 1800s, when people started using microscopes aggressively, some scientists thought they could see miniature humans packed inside sperm heads. At the time, that view was not unscientific. It was based on the available tools that the scientists of the time had. Currently, genetic analysis has revealed that the interactions which take place between genes and the organism are not as simple as once thought. Interactions are much more complex than they were envisioned in the past. However, there are still some characteristics that are under very simple regulation, where one gene controls alternative phenotypes. That is the case with the ability to taste a chemical PKU. Either you can taste it or you can't. There's no middle ground. Students are therefore warned to be on the lookout for more sophisticated genetic interactions. The father of genetics is clearly Gregor Mendel, who lived in the 1800s, and he published his work in 1860, but that work was ignored until the 1900s. Indeed, his manuscripts were sent to Darwin to comment, and later on we learnt that Darwin did not even open the packages that he received. Gregor Mendel was considered a second-class player in the field of biology. It was with the rediscovery of his work that we truly gained an insight into this genius. Gregor Mendel was meticulous in the way that he did things. He is noted for his experiments on the garden pea plants, although he did work on other organisms as well. His notebooks, which are still available, track his every experiment. Essentially, he studied 7 characteristics, 7 traits, as they're called in genetics, of these pea plants. And they're illustrated here at the top of this row. So he studied the shape of the seeds, the peas themselves. They could be wrinkled or smooth. He also studied the color of the peas. So in different strains of pea plants, the pea seeds could be yellow or green. He studied the color of the flowers, which could be white or purple. He also studied where those flowers grew on the plant themselves. Some of them were axial and some of them were terminal. He also looked at the shape of the pea pods. Some of them were inflated and some of them were pinched around the seeds. And he also looked at the color of those seed pods too. They ranged from being green or being yellow, nothing in between. And then he looked at the size of the plants themselves. Some plants were really tall, other plants were really short. Mendel's experiments are nicely chronicled in his journals. His first genius was to make sure that when he bred plants together and produced offspring for a particular character, he got rid of any plants that produced alternative shapes or sizes. For instance, when looking at the yellow pea color, he kept breeding his plants again and again for many generations until all the offspring at the end were producing nothing but yellow pea seeds. And he did the same thing in a different part of the garden where he bred the green colored pea plants. He made sure that the plants that he used were clipped so that the pollen of one could not accidentally in the wind be transferred to the stigma of another. The resulting strains are called pure breeding because they produce nothing but that particular characteristic. When he bred on purpose the parents of yellow pea plants with the parents of green pea plants, he observed in the next generation that all his plants were yellow seeded. 100%. Had he stopped here, there was not much to deduce that others before him had not deduced. His genius came when he took these F1 plants, and F1 stands for the first filial generation. When he took these plants and he self-fertilized them with a paintbrush, that he waited for the next generation to appear. And in the next generation, F2, the grandchildren of the parents, he found that there was a mathematical relationship between the numbers of pea plants that contained yellow seeds to green seeds. And the ratio was always 3 to 1, or close to that with an experimental error. No matter which of the 7 traits he studied, he always got the same 3 to 1 ratio. And that got him interested in examining further what was going on deep inside these pea plants. He applied his knowledge of mathematics, which he had gained at university before becoming a monk, and came to some nifty conclusions. In particular, he came out with a law of segregation to explain how factors, as he called them, were being combined and segregated during the production of gametes and the formation of the next generation. He also came up with the naming convention for these factors. So he used a capital letter for a dominant factor and a lowercase letter for a recessive factor. We call those now alleles. So the big Y will be the dominant allele and the small y will be the recessive allele. We don't know which allele does what until the results of the F1 reveal the relationship. So in this case, when he bred the yellow and the green together, he had no idea which of these two would be dominant until he saw the actual progeny in the F1. And the appearance of yellow indicated that yellow was the dominant allele and the green was the recessive allele. The only way he could explain his findings was to assume that each parent had 2 letters. In this case, a big Y and a big Y. But the gametes that the parents produced only carried one or the other of those 2 factors. But the gametes only had space for one factor or the other. It didn't make a difference here in this first cross because big Y, there was no choice. The gametes all contained big Y. And likewise with the green parents, the pure breeding green parents, they only had small y. So all their gametes, 100%, had no choice but to be small y. But when the gametes came together upon fertilization, the big Y and the small y came together in these individuals. So all the F1, the children, were big Y, small y. When the F1 were bred together, the gametes that they produced, whether they were male parts or the female parts, had to contain just one or the other of these 2 alleles or these factors. So some of the eggs got the big Y and other eggs got the small y. And the proportion in which the eggs received those factors was 50-50. So half the eggs were big Y, half the eggs were small y. Likewise, the same thing happened in the male parts of the plants. Big Y, 50%, small y, 50%. And if this egg was fertilized by this sperm, then this table here, known as a Punnett square, easily shows you the offspring that will be generated. So here, 2 big Y's will come together and they will produce a yellow seed. Likewise, here, a big Y sperm and a small y egg would come together to give you this heterozygous condition. But because the big Y is dominant to the small y, all these offspring would also have yellow seeds. Likewise over here. But the last row here, or column, this is a Punnett square, the small y eggs were fertilized by the small y sperm. So these offspring that will be generated would contain small y. And just by looking at the number of boxes out of the total, out of the 4 boxes, one set, 1/4 or 25% would be the small y condition. Therefore, the ratio is 3 yellows to 1 green. Thus, we have 2 segregation events going on. One right here, when these gametes are produced inside these parents, and once again when these gametes are produced inside these parents. And if this process was to continue, the F2 generation, these ones here, would then produce their own gametes, and that would be another segregation event. So segregation occurs every time meiosis takes place inside these plant cells. The way these factors or alleles move through organisms is the same for all sexually reproducing organisms, irrespective of the difficulty with which the character can be studied as genetics. So it doesn't matter what you're studying, height, intelligence, which are very complicated genetic components, or you're studying simple things like taste. Everything is based on genes moving through organisms into their gametes and then upon fertilization into the offspring. So breeding of dogs, which has been taking place for many, many thousands of years, unknowingly was manipulating these factors according to which were beneficial or liked by the human masters. With respect to humans, another example is mentioned other than taste, where we can now follow how in parents that characteristic or that trait is passed through into subsequent generations. So applying the same knowledge from the pea plants, we can, for instance, this albino man, albino simply means a condition where no pigment is produced, so the eyes are pink, the hair is blonde, and the skin is very light, and normally pigmented individuals. So this albino man, who is diploid, therefore carries 2 chromosomes, and on each chromosome there's a small a and a small a at that location, breeds with a woman who is normal, then their children will all be normal because the big A is dominant to the small a. So the big A makes the pigment, whereas the small a has no effect. All their children would be pigmented normally. But if they were then to be crossed by themselves or mated with another family that contains a similar genetic profile, i.e. heterozygous and heterozygous, then their offspring, the second generation, the grandchildren of these parents, they would be producing a 3-to-1 ratio. By studying humans across many, many families who have undergone this type of genetic passage, then the ratio would indeed be 3-to-1, but maybe not for the same one family. That's the same thing as having families that have 5 children and all 5 children are girls. Technically, every time they have a child, there's a 50-50 chance that it will be one sex or the other. For ethical reasons, human experiments are frowned upon by society. Therefore, the best alternative option is to study human pedigrees that already exist. For instance, there could be a child, a male here for instance, so boxes that represent males and circles represent females, and colored boxes, they represent persons with that particular condition. It could be a disease or just a condition. And open boxes like this, or open circles, represent those individuals that don't express that particular condition. The lines indicate marriage, and reproductive success is indicated by these lines branching downwards, leading to the production of children. And the children are always indicated with the first child coming off first on the left-hand side, and the second child, if there is a second child, coming off subsequently. So let's look at a couple of these. So let's look at this marriage here between 1 and 4 in the 3rd generation. This male married this female, and they were successful in having 5 children. And the firstborn was a female, normal. The secondborn was a female that was deaf. The thirdborn was a male who was deaf. The fourthborn was a female who was normal. And the last child was a normal female. Likewise, 2 and 3 had 5 children, 2 males and 3 daughters. All 3 daughters were afflicted with deafness. A pedigree analysis can be applied to any organism that breeds sexually, including dogs. The genius of Mendel had not finished yet. What he did next, once all his experiments looking at single characteristics were finished, he moved on to look at 2 characteristics at the same time, 2 traits at the same time. So he combined all his traits together, all 7 traits. And in this example here, he's not only looking at the color of the seeds is also looking at their shape. So we have round seeds, then we have wrinkled seeds. We have yellow seeds, and we have green seeds. The initial parts of his experiments were the same. He had pure breeding lines of yellow, smooth, let's call it round, seeds, which he bred for many generations. He did the same thing for the other parent, which had green and wrinkled seeds. Once he was happy that he had pure lines, he purposefully bred these with these, so the yellows with the greens. And he waited until the next generation was produced, the F1. And as he expected, all the seeds were of the dominant type. In this case, as we know from a previous example, yellow is dominant to green, so we expect the seeds to be yellow, and they were. But the question is, was wrinkled dominant to round? No, it wasn't, because all the seeds were round. So that tells us that round is dominant to wrinkled. So we can use appropriate terminology to refer to those genes. So the allele for roundness is given the capital letter R, and the allele for wrinkled is given the lower letter R. We could have chosen any letters we like. We could have chosen W. As long as we know what we're talking about, that's okay. Then Mendel did the same thing. He took the F1 plants and he bred them together with themselves, self-fertilized. And this time, he expected to find a ratio that was different to the 3:1. And indeed, he got a ratio of 9:3:3:1. Yes, 4 ratios. And it's possible to have multiple ratios because it's just a relationship between the different categories. So some of the seeds were yellow and round. Some of the seeds were yellow and wrinkled, and you can see them here. And some of the seeds were green and round here. And the last category were green and wrinkled. And if you look back, you can see that each gamete, each egg, has to now contain a copy of each gene. So big Y, big R. But it can also have big Y, small r, or small y, big R, or small y, small r. That's because the chromosomes are being segregated into these gametes, as we learned through Mendel's first law. But what Mendel deduced from these experiments, because the ratio was always the same, doesn't matter what he tested, he came to the conclusion of the law of independent assortment, which states that if you are studying 2 different genes, then each gene pair will have its alleles independently go into the gametes, irrespective of what the other allele was. So what that means in layman's sense is that if the allele for yellow goes into the gamete, there's no reason why the round must always go in with it. The wrinkled could go in with it. So you're now mixing things up. Please understand Mendel's first and second law in respect of our learning of the cell cycle in the last chapter and earlier in this chapter. And that's exactly what the book does next. It takes modern biology and it applies it to what Mendel was seeing. So if you look carefully here, we have different chromosomes in the pea plant which carry the different characteristics. Based on the last slide and the example given there, we're going to use the same terminology here. So the large chromosomes in pea plants contain the gene for the shape. And these 2 small chromosomes, they contain the gene for color. So remember, Y's were used for color and R's were used for the shape of the seeds. So if you follow through here, you should be able to see how the gametes are formed within one of the double heterozygous parents. These are the F1s. So the F1, in this particular parent, its chromosomes could line up in either orientation. You can see here the top half is the same, where the small r lines up to the right and the big R lines up to the left. However, they have no control over how the other pair, the smaller pair of chromosomes will line up. In this case here, we can see that the big Y has lined up to the right, sorry, to the left, and the small y has lined up to the right. Or alternatively, they could have lined up this way. So this is where the randomness comes in, this independence in the way that the chromosomes line up. And from this point onwards, the way that the spindle works is that the chromosomes here on the left-hand side should always be pulled to the left, and the chromosomes on the right, that homologous pair, should be pulled to the other pole. And that will result automatically through the second round of meiosis in the final haploid cells. So if you look at this half of the figure, this is the result of the 4 types of haploids you get from this meiosis 1 cell. Or alternatively, if they're lined up like this, then you will get these 4 alternatives. But if you look very carefully, you should be able to see inside a single parent there are hundreds of thousands of gametes that are produced, and they can line up 50-50 this way and 50-50 this way. So in a population of these individuals, you can end up with all versions of these gametes. So please understand that. There is one piece of luck. Yes, this story has a piece of luck. When Mendel was studying his 7 traits in the pea plants, he was lucky that of the 7 traits he studied, none of them were of characters that were next to each other physically on the chromosomes. Why is that a big deal? The answer is something to do with linkage, genetic linkage. This slide from the textbook tries to illustrate that. Imagine that you're looking at these particular genes, a gene that controls something over here, another gene that controls another character over here, another gene that controls a different character over here, and a last 4th gene that controls a very different character over here. Because of crossing over, the distance between these 4 genes is so great that on average you are going to get crossing overs taking place in these arms. Therefore, these letters, the big F and the big E, can become mixed with the small f and the small e crossing over and residing on that chromosome. So that would be equivalent to having F on a different chromosome. If you look down here with A and B, A is controlling one characteristic, B is controlling a different trait. These genes are so close together, in fact they can be closer than this, that the chance of having crossover between them is reduced because of their closeness. And that will prevent small b from moving away from small a. So this part of the chromosome would be passed on to the gametes as it is, without crossing over. And that's what linkage is all about. So genes that are close together on the same physical chromosome tend to stay together unless moved by crossing over events. But because crossing over depends on distance between the two genes close together will rarely have a crossover and will tend to be inherited. Their alleles will tend to be inherited in the combination that the parents had them and seldom recombined. So crossing over scrambles genes on the same chromosomes. How are geneticists able to tell if 2 genes are close together and they'll have some kind of linkage? The answer is by looking at the ratios. If you're expecting a ratio of 9:3 to 3:1 between 2 different traits, and that ratio doesn't materialize in the F2, then there's something going on with the distance apart of the 2 traits, and that's an indication of linkage. Let's move on to a different concept. Let's apply how genes influence the shape of proteins, which we learned in other chapters. So assume that this protein here in green has a particular shape and an active site based on the information carried by its gene. And through transcription and translation and protein folding, this protein is generated. A mutation in the gene itself could alter the protein in ways indicated here in panels B and C. Panel B is the most obvious in that the mutation could knock out the production of the protein completely, for instance by putting a stop code on, at the beginning of the messenger RNA. And if that happens, the protein will not be produced. So we have a loss of that protein inside the cell. Therefore, that's considered a mutation that leads to the loss of something. And in this case, the function of that enzyme will be lost. Therefore, these mutations are called loss of function mutations. But that's not the only scenario. We could have a mutation that only affects one amino acid amongst all the others, and that for some reason happens to be here at the active site, and that reduces the function of that enzyme. So those are also classified as loss of function mutations. And indeed, we could have another premature stop codon, but further along the gene, that allows the polypeptide to be made, but not completed. And that will knock off the active site completely, and that will also be a loss of function mutation. So those loss of function mutations are the most common things to happen, depending on where the mutation is and how the protein is manifested. There is another possibility too, as indicated in panel C. This is called a gain of function mutation, sometimes hard for students to perceive. Imagine that a mutation takes place on the DNA, and for some reason it makes the active site more aggressive. Therefore, it binds to its substrate much more readily and promotes enzymatic reactions even faster. So there, the function of that protein has been elevated. It works faster. And you may think that's a good thing, but it doesn't necessarily mean it's a good thing. Sometimes the gain of function increases the biochemistry so much that it can upset other balances within the cell. And we'll see examples of this in the last chapter, chapter 20, when we look at cancer cells. Looking at our genomes, the genome is the complete set of genes that we carry. We all have different genomes. So I have the perfect genome, of course, and so do my TAs, as it says here. But the rest of the world, other than the three of us, you folks are imperfect. And each of you carries, on average, 6 to 10 major genetic issues which will crop up sometime during your lives, if they haven't already. And those issues sometimes can be treated medically, and other times they cannot be. But regardless, nobody is perfect, apart from, of course, as it says at the top here. So the way that genes work in populations is a fantastic opportunity for us to see how nature works. So priority is given to the population and its survival rather than the survival of an individual. So you and I really don't matter as part of the human species. What matters are the genes that travel through us into the next generation in this particular environment in which our species is currently living. So nature has mechanisms that it tries to keep bad alleles from becoming too prevalent in the population and destroying our ability to survive. And it tries to promote good alleles which help the species survive in that particular environment. But nature is not perfect and it cannot predict the future. So it does what it can with what it's given. So deleterious mutations reduce reproductive success in terms of evolution and are constantly being eliminated from the population. But mutation is responsible for introducing new mutations into the species, as well as migration from other populations. So bad alleles can linger in the heterozygous condition or in individuals that have a reduced expression of that particular disease. And that's something that you'll have to take up with a genetics class. There have been two approaches to studying genetics. Initially, there was only one method and tools available, and that was the classical way of studying genetics. There, scientists had to wait for mutants to be brought to their attention, or they performed experiments where mutations were purposefully introduced using mutagens, substances that induce mutations in the DNA. And then they would study those mutations or mutants and try to uncover which genes were responsible and where those genes lay on the chromosomes of that species. But since then, our technology has improved, and now we perform what is called reverse genetics, where we begin with the DNA sequence of a species, we sequence the DNA, feed that into computers, and then compare that data with other creatures or within the same species to seek the genes as they lay on the chromosomes. And from that, determine how mutations could be generated by predicting the structure of RNA and proteins. So there are two methods, and this one is taking over more and more from the classical method because of time savings and, of course, technological advances. In your textbook, they have these colored pages where they introduce or reinforce many of the words that geneticists use constantly when communicating information. Please learn these words, and there is a glossary at the end of the book where you can go and get definitions of every single one of these words to your satisfaction. Genetic mapping, as we just mentioned, is a way of measuring the rate of crossing over to determine the distance between two closely located genes on the same physical chromosome. And the term centimorgan is used to indicate how far apart two loci means the home of a particular gene, how far two genes are from each other. And that was first deciphered by Thomas Morgan, and therefore in his honor the word centimorgan is used, but it really is a percentage, how far apart the two genes are. So the smaller the number of centimorgans, the more closely the two genes reside on the same chromosome. How do you determine that? That's determined just by looking at the offspring produced of different types and then putting that into a simple mathematical equation. So let's delve into the classical genetic analysis method and give you a perspective on that. So one way to generate more mutations, as I just mentioned, is to use mutagens, chemicals that induce DNA-based changes. Now, whether you use a mutagen or you're looking for naturally occurring mutations, the outcome is more or less the same. So this chart here shows you the top half, but there is another half of this chart that's not mentioned in this figure, but is talked about in the book. Regardless, let's just concentrate on this. So what type of DNA changes can you witness with mutagens or mutations? Well, the simplest is this, where a particular base, a G, is changed to another base, in this case a C. So we call those nucleotide substitutions. So substitution. Just like in football, one player comes off, another player takes his place. But the number of players on the field is still the same. An alternative thing that can happen, indicated here, is that you could add a player to the field without taking anybody off. And that's called an insertion. So this nucleotide A has found its way between the G and the C of the natural gene. You can also remove a player without substituting that player. So here the G has disappeared. So in both cases, there's been an insertion and a deletion. And insertions and deletions are called indels in the genetic community. So in a paper, you may say, oh, this gene is susceptible to indels. That means insertions and deletions. Of course, there's nothing to stop you from adding two players to the field or taking two players off, or more than two players. So this is basically showing you a very simplified view of how the size of a gene can be changed in these four parameters. Of course, there's other ways. You can completely duplicate a whole section of a chromosome that contains thousands of genes. Indeed, you can copy a whole chromosome, like in trisomy 21, Down syndrome. And in other cases, like plants and amphibians, you can copy the whole genome. All 40 chromosomes could be duplicated 3, 4, 5 times. So you can see this is the bottom of that spectrum. The screening processes have been pretty rewarding. So the genetic screening can result in the discovery of creatures that are particularly relevant to the study that's been taking place. This is illustrated here very nicely. These are small tiny worms about 1 millimeter long. You can barely see them with your eyes, but they live in the soil, and these nematode worms are exhibiting over here social behavior, where here they are loneness. So if you were studying a gene for social behavior, then these mutants that don't want to associate with other worms would be ideal candidates to study to see which gene is broken, stopping them from coming together, as indicated here in this social gathering. Very powerful stuff. Indeed, the nematode worm is such a wonderful model organism in which to study development, because it always has 959 cells in the adult, in a normal adult, and it's transparent. So no wonder it's used for developmental analysis. Applying a recent discovery called RNA interference, RNAi, to the nematode worm has led to a wonderful system where we can determine the effects of any gene being shut off individually. So if some of you remember in Bio 2, we watched that video about RNAi from Nova Science Now. If you need to refresh yourselves, please watch a small segment of that video. In this case, every single well contains bacteria that have a double-stranded mirror image RNA introduced into their DNA. So the DNA of the bacteria is technically manipulated, and then the worms are introduced into these wells, and the worms will eat the bacteria. And as the worms eat the bacteria, they consume that RNA, which enters their cells, and that triggers the RNAi response using their risk proteins. So the risk system neutralizes any similar genes that the worm itself is producing, therefore shutting it off. And that way the scientists can then observe which worms are phenotypically changed as a result of that particular gene being turned off. So in this example here, in a well that contains a particular RNA, the worms are made sterile by the presence of that double-stranded RNA. So the gene that corresponds to that RNA must be responsible for sterility. The next major concept that students need to learn is something called conditional mutants. Conditional mutants are mutations, normally of proteins, sometimes RNA, that manifest themselves under certain conditions, maybe under certain pH, or maybe salt conditions, but most commonly under different temperatures. So here we have a set of yeast being grown on a plate, and then we have replicas of this plate made. So these are two identical replicas containing the same colonies in the same places. And if you put one plate in an incubator at 23 degrees and its sister plate at 36 degrees, you can notice that this colony here is not growing at 36 degrees. So that colony contains a set of organisms, yeast cells, all identical clones, which are temperature sensitive. So they're called temperature sensitive conditional mutations. So why do we want to study genes that are temperature sensitive or conditional? And the answer is, maybe that gene is necessary for the growth of the colony, and it won't allow the colony to grow if it was mutant. But we want to now change it to the mutant version simply by raising the temperature, and then we can study what happens as a normal gene and a mutant gene. So this takes some getting used to. The next concept is the complementation test, another hard one, but not really. If you look at it over here in this figure on its left-hand side, imagine that we have two genes. It doesn't matter where they are. In this case, they're very close together on the same chromosome. They don't have to be. Suppose gene A controls the shape of the fish, but somebody else has also found the same fish in the same shape, and they're not sure if there's another gene, but assume that there's another gene B that's responsible for that. So gene A or gene B, either, if broken, will result in the fish having this weird shape. The question is, how does the scientist know whether this shape is caused by a mutation in one gene or two genes? Well, the answer is simple. You take these parents, you breed them together, and you wait until their children are born. And if the children are born in a normal configuration, then each parent must have had a defect in a different gene. To prove that, we can actually take a gene that has mutations at different points. Remember, a gene is a string of nucleotides, and you could have a mutation at the beginning or the middle or the end of that gene. In this case, on the left-hand side here, A1, the mutation is near the beginning of the gene, but it still produces a defective protein that makes the fish shaped like this. And you have another mutation later on in the same gene that also affects the same protein, making the fish, of course, the same shape. So if you breed these two parents together, then you'll end up with all their offspring having the same shape as the parents. That's because both these genes, the same gene, is broken. And what happens in the offspring is that they inherit the same gene broken in different places, but it's still the same gene. So here, the two genes have complemented each other. They've helped each other to overcome their handicaps, and they end up with a normal creature. So there is complementation. Over here, the two genes don't help each other because they are one gene, and there's no complementation. So but that scientists in the past had no way of looking at the DNA. This is what they used to do, and still can do. There's a real-life example of a complementation test that was tried in the past. So once upon a time, we had this species of bird where people found white feathers. And the question was, is the white feather controlled by one gene or two genes? Well, what they did is they had a male bird and a female bird who happened to have white feathers, and they bred them together. And their children all had normal feathers. So that told us that complementation took place. Therefore, the mutation here was in one gene, and the mutation in the female was in a different gene. Think about that, please. The speed with which technology is moving currently is absolutely frightening and amazing. When I was young, it took 10 years for the Human Genome Project to conclude, and it cost about $3 billion to sequence the first set of human genomes, using the best technology available in the 1990s. Today, using current day technology, 2018, we can do the same job for about $400 in as little as a couple of hours on one of the newer machines. In fact, there's now an add-on to a smartphone that allows you to sequence within an hour or two the genome of any creature in the wild. Amazing. If you wish, you can have your own genome sequenced by sending it to one of the companies like 23andMe and specifying that you want the whole genome sequenced. Or you can ask them to sequence maybe 200-300 genes that are normally problematic in the human disease set. Regardless, do you really want to know what lingers in your DNA? And if so, will your healthcare company want to have the same information? Another thing that we have learnt over the last few decades is that humans, individuals, we carry mutations on our DNA that are present in other individuals in the population. If a particular location on the DNA is present in a variant form in 1% or more of the population, then that has a special name. It's called a polymorphism. A polymorphism. And that's indicated here in this particular diagram. So individual A, that could be you, individual B could be your friend, individual C could be me, and individual D could be one of the TAs. If we were to sequence that exact same location on our DNAs, it's possible that you may have a GC at that location, and your friend may have an AT, and I may have an AT, and then the TA may have a GC. But if this is present in the population as a whole of all humans, or that population, say North American Indians, then that's called a polymorphism. And these polymorphisms can be used to our advantage. If they occur at the same spot on the DNA and do not involve their neighbors but a single nucleotide, it's called a single nucleotide polymorphism, SNP, or SNP. These SNPs can exist in many locations around our DNA. Remember, we have 3 billion of these locations, 3 billion, which is huge. So having 300 million locations that differ is not unusual. So humans can have 300 million locations that differ in this way. And those differences can be used, as we'll discover in the next few slides. But before we leave this slide, it's important to understand that the human species went through a bottleneck. A bottleneck is a constriction in the number of individuals that are breeding and surviving. So about 200,000 years ago, the human species dwindled down to as few as 10,000 individuals. And we are the children of those 10,000 individuals, all of us, all 7.25 billion of us on the planet today. Therefore, we do share some DNA of our ancestors from that time. And because mutations are pretty rare events and crossovers are pretty infrequent along our huge chromosomes, the probability of large regions of our DNA still being present today in the same orientation as in those ancestors is quite large. And we call those haplotype blocks. So this region from here to here could be millions of base pairs long, and it may still contain the same arrangement of these particular SNPs that our ancestors had. So using that information and similar types of information allows us to look back into the past and come up with some fantastic observations. For instance, it appears that the human species evolved in central sub-Saharan Africa. And here is where our ancestors first migrated out of Africa from. And they left Africa and went in all different directions. So some went south into southern Africa, across the Great Plains and the deserts down here. Some went towards the coast into Somalia and Ethiopia. Others went across into the jungles of the Congo. And others went across, which was at that time a rainforest, into the Arabian Peninsula to populate the planet. And that first wave of migration, they became the Neanderthals. So the question is, where did Neanderthals come from? They were evolved from the first wave of humans leaving Africa, or humanoid-like apes. Then, more recently, for some reason, there was another wave of human migration, which took place about 100,000 years or less. And these second wave of humans, when they left, they met our first wave of ancestors. And between the two, there may have been some conflict and some interbreeding. Eventually, all the other subspecies or species of humanoids went extinct. And we now today have a single species of humans on the planet, which is in itself amazing. We can also, by looking at DNA extracted from the teeth and bones of Neanderthals and other humanoids, we can now see that some of us, particularly those that left Africa in the second wave, possess about 2 to 4% Neanderthal DNA. The purest humans alive today are those that evolved in southern Africa, who never came in contact with Neanderthals or any other species of humans. The last thing we want you to know with respect to polymorphisms is that we now know, through a massive effort in the 1990s and the 2000s of mapping where every single SNP resides on the human chromosomes, as well as other types of variation, we can use that data to co-find genes. So if you look at this figure here, that tries to illustrate that. Imagine that these are polymorphisms, that there's one variety in a particular human being, but in the population there's two types. In this case, they could be red or blue. And you can see here, in healthy individuals, there's no real pattern as to which one the healthy individual contains, unless you get down here. Here, everybody has a blue marker at that location. Likewise, in affected individuals, the same randomness exists on chromosomes either side of this dark gray area. But within the dark gray area, everybody that's affected with that particular disease has the alternative form of the SNP. By looking at which SNPs are present in which population, we can then use those SNPs to narrow down onto the gray area and then sequence the gray area looking for any regions that may contain genes. That way, this helps us hunt down where to look by using these landmarks. If you have no idea where the gene resides, you can look at all the markers that are in the database and try to correlate which ones are present in affected individuals versus normal individuals. Because you're looking at the entire genome, that is called a genome-wide association study. Genome-wide, because it's the entire genome that you're looking at. Association, you're trying to find relationships between those markers and the diseased individual, and that's a study. So this is a very common term that you find in the literature these days as to this methodology. Lastly, the chapter talks about how all the analysis is pointing in a particular direction when it comes to disease and individuals. Unfortunately, the GWAS studies, they only indicate a doubling or tripling of a risk factor for that individual. So if you have a marker for diabetes that's associated with a landmark, the chances are that your risk of diabetes is only increased by maybe twice if you have that particular zone of chromosome on your DNA. That is not what we were expecting. What we have discovered since is what this slide is telling us. It appears that most of the major serious illnesses that humans are afflicted by are the result of mutations that happen that are unique to our parents and us. So these are de novo mutations. That means new mutations that have happened in the germline that led to you. And that germline passed through your mother and your father. So even though your mother and father may not have that predisposition to that disease, their mutations that they had in their testes and their ovaries, which seem to be quite prevalent, were responsible for giving you your profile. So sequencing of family DNA reveals this more recent finding to be true, that this near de novo mutation rate is responsible for the majority of our risk factors for developing something. So a subtype of DNA sequence called exome sequencing, so remember introns and exons, well, this is based on exons. So exome sequencing is where you focus on sequencing just the RNA. And that will give you an insight into which types of proteins that individual is making at that time in that tissue. And that has the most useful correlation between these serious diseases and this type of mutation. This is amazing. But this work is still in progress, and I think we'll end up with a more solid understanding of how our diseases, our personal diseases, are justified. Thank you so much.